Okay, hello everybody, so let's get started. It's my great pleasure to welcome here Professor Jennifer Apple from the University of New York, from the sunny Geneseo College. And the reason why Jenny is here is not because she would come for Europe for a single day or something, but actually Jenny is staying at our department in my lab for the next 3 months, so you will be able to meet her later if you want. Jenny, she's an ecologist, and she has been studying different topics. In the past, she has been studying, for example, herbivore and plant interactions on the Mount St. Helens. That's a famous volcano where lots of research has been done, mainly in the 70s. Later on, then she switched to, for example, to interaction among ants. And the topic that she chooses today is about spiders. However, not a...
Surprise, surprise!
Yes. Spiders which actually imitate ants. So I hope you will enjoy it, and here we go.
All right, thank you very much. And thank you, Stano, for hosting me in your lab this semester. I look forward to our collaboration. And I also thank the Terrestrial and Vertebrate Ecology group for sharing their office and lab space with me this semester during my sabbaticals.
Maybe you could speak up.
Okay. So, I want to tell you a little bit about where I'm from. Probably none of you have heard of Geneseo, New York. I live in western New York, although I was actually born and raised in the western United States in New Mexico. And this is a picture of the state of New York, and with the Czech Republic outline superimposed, just to give you an idea of the scale of the state. I'm nowhere near the city. I'm up here in Geneseo, which is on the edge of the Finger Lakes, which are all of these lakes that were formed by glacial activity as the glaciers retreated across New York State. You might have heard of some of these other places like Rochester and Buffalo, which are nearby where I live. And western New York is very rural. It's got a lot of rolling hills. This is actually where I live. It's on kind of top of a hill, a drumlin that was formed by glacial activity. There's a lot of agricultural fields, dairy farms, forests in various stages of succession. And I live only a 10-minute drive away from the State University of New York in Geneseo. This is our campus. About 5,000 students there. It's primarily an undergraduate institution. There's just a few master's programs in education and business. There's a Genesee River Valley over to the west of our campus. And just to the south of our campus is basically where most of my field research has been conducted. In our arboretum, our 8-hectare secondary successional forest is where I do a lot of my field work for my research and also working with students. And that's where I've been working on ant-mimicking spiders. In particular, I work on a species that's in the family Salticidae, the jumping spiders. These might look familiar to a lot of you. Typical salticid spiders look like the page on the left here. But the salticid spiders that mimic ants are very different in form. Their body shape is very different, modified with constrictions you don't typically see in typical salticids. The coloration is changed. They're much less hairy. So there's a lot of modifications they've undergone to resemble ants. And sometimes they are really quite faithful to the ant species that they mimic. For example, this species, which is actually in the same genus that I study, Myrmarachne, looks very similar to the model species of ant that it is mimicking, a pseudomyrmex ant. Here's another mimic from India that looks very similar to the chromatogaster ants they are in association with. Not all ant-mimicking spiders, though, are necessarily this faithful to any particular model species. Sometimes we don't know exactly which species of ant they're modeling, if it is any one in particular. And they can even, over the course of their development, resemble different ant species as they change their form. So the species I study is Miramaracity Formicaria. It's a relatively new arrival to North America. It was first noticed in Ohio around 2001. It's actually native to Europe and Asia. Other observations were made shortly thereafter in New York and in Pennsylvania, also in Ontario, Canada. Here you can see that it has striking sexual dimorphism. The male is on top here with these very enlarged chelicerae, whereas the female lacks those enlarged chelicerae. So they're easy to tell apart as adults. And it was actually European Spider of the Year in 2019, which is very exciting. So ant-mimicking spiders not only look like ants in their form and color, they also behave like ants in many ways. Their locomotion is often very ant-like with their meandering paths and the way that they move through space. And they also will raise either their first or their second pair of legs like their antennae, which is pictured here. And this is from a study, one of the few actual studies on Myrmarachity Formicaria done by Paul Shamble, who showed statistically how they were similar, their movements are very similar to those of ants rather than to those of non-mimicking spiders. And I'm going to show a little video that illustrates this movement. This is a female. You can see how she waves that first set of legs out in front of her. Mainly when she's stationary, actually when she moves, they're actually always on the ground, but as she stops, she lifts them up and waves them. So there's not actually a lot of study of Myrmarachne Formicaria in its native range, at least in the published literature. One of the few studies is actually by Stanley Picard and Giros that proposed that a possible model ant species for this spider is Formica rufibarbus. Not only does it have a similar appearance and shape, but it also has some chemical similarities as well. Similarities that cause the ant to respond differently to this spider than to other non-mimic spiders. Some other people have suggested perhaps Myrmica rubra. The European fire ant might be a model. There are some people in Ontario where Myrmica rubra is actually quite common and invasive have suggested that. We know a lot about kind of the way we know how Myrmarachne Formicaria has spread over time is thanks to people observing it, and mainly citizen scientists observing it around their homes and businesses over the past 10 or so years. I don't know if you've heard of it, but iNaturalist is a citizen science platform. It's a website and mobile app that people can use to take pictures of pretty much any kind of form of life, plants, insects, mammals, birds, fungi. They can post these observations to the website. They can get some artificial intelligence help with kind of narrowing down the identification, and then actual experts and knowledgeable amateurs will weigh in on observations on this app to help identify people's observations to a species level to which we can have some confidence. And there's actually been 605 iNaturalist records posted in North America since 2008. Mostly since 2012 is when they really started starting. That's compared to 81 in Europe. That just really reflects the popularity of this web app in North America. But this map is showing how they've kind of spread over time. This is showing early observations between 2008 and 2017, and then kind of spreading out from Ohio, where they first noticed, the Buffalo area, Toronto, and further out into New York, Ontario, Pennsylvania. There's even some seen in Chicago recently, out in Vermont. There was one recent one in Massachusetts. And just yesterday, someone found one in New Brunswick on that border with Maine, which is really far out there compared to all the other observations. And I looked at the picture, and it's definitely Myrmarachne. This is actually a more up-to-date map that shows the distribution of the species. So it's spreading steadily throughout northeastern North America. So how did I come to study this species? I'm actually fairly new to spider research. I've been spending the last 15 years, I guess, focused on ants on our, in that area campus I mentioned before. I study these Formica ant colonies, or mound nesting ants like this, and particularly this kidnapper ant species called Formica subintegra, which raids colonies of another ant species, in this case, in my site, Formica glacialis. It enters the colonies, it kidnaps the brood, mainly pupae. They bring them back to their own colony, and when those pupae mature, they become the workforce of the kidnapper ant colony. And they do all the foraging for the ant colony, all the nest construction, brood care, pretty much everything. The raiders only go out and raid for more workers. So I've been tracking the raids of these ants every summer for a long time, checking colonies, looking at ants. So I'm always looking at the ground at the ants. And occasionally I would notice this sort of ant-like thing, but it didn't quite look right, and notice that it was actually really a spider, the Myrmarachne Formicaria. And then I started noticing that they were actually using these flags, these vinyl flags that I put next to my colonies to build their silken shelters on. So they were nesting in the flags right next to the ant colonies. So I started to wonder if they're actually associated with the Formica ants in some way. And in 2017, I really started to study these in earnest. Here's a video that kind of shows them among some ants. So this is actually a video where a kidnapper ant colony has been on a raid and is coming back with loads of pupae to its own nest. Let's skip ahead a little bit. You can see this is the female ant-mimic, and around you can see below here there are ants moving around. They have white pupae. There's an ant. So she's just kind of wandering around on this layer with ants coming and going around her. Always moving. I've never seen them actually enter nests, go inside the nest, but just kind of they'll be in the vicinity of these ants. So the original reports of this spider in North America were mainly from human-made settlements. If you look at the first publication, basically they're describing they found it on a wall, they found it in a coffee cup, they found it walking down the linoleum or descending from the ceiling in a warehouse, not in any kind of natural setting. Even the pictures you see on iNaturalists are in containers. They're not generally on any kind of natural substrate, except this one right here. And so I began to wonder, what is it actually doing in these natural habitats, since all of the reports of it are associated with these human dwellings and heavy areas of human activity. But I was clearly seeing it in my natural field site, where I studied ants. And so in my lab, our goal is to try to characterize the natural history of Myrmarachne Formicaria in its invaded range. And so we're investigating things like patterns in space, the degree to which it actually does interact or associate with ants, its habitat preferences, what kind of habitats natural habitats it lives in, patterns in time, trying to describe the phenology of the species, when do males and females appear, when do different reproductive stages appear, their reproductive activity. Also characterizing their behavior. I've noticed interesting male-male display behavior in the field. Also interactions with potential enemies, given their appearance, their resemblance to ants. Their overwintering habits, given that we can have pretty cold winters in New York. And then finally, their genetic and phenotypic variation. So to answer some of these questions, some of the survey methods I've been employing make use of the flags that they like to build their nests in. And so one of the questions I was interested in is if they do actually preferentially nest near ant colonies. So I used existing flags that were marking nests, flags on kidnapper ant nests, flags on nearby host nests that might be exploited by the kidnapper ant, and a control flag with no ant nest next to it. And I would check these flags 15 times throughout the summer to see if any spiders were occupying them, and had a bunch of different sets of these trios. I also set up grids of these flags in different sites to try to see where the spiders were more common, what kind of habitats they were more likely to occupy. These grids consisted of 12 flags, alternating blue and yellow, about 1.5 meters apart. I had 18 plots in 4 different locations around the town of Geneseo, and I checked these weekly in summers of 2019 and 2020. Actually, I'm continuing to do this. I did it in 2021, but I haven't analyzed those data yet. I noted the presence of spiders, the sex, and the stages, whether they were producing eggs, spiderlings, and also if other spiders were present as well, because other spiders will also use these flags to build shelters in. In 2019, I specifically sampled ants in all of these plots, and I interspersed between these flags. I would set up either, I would have two methods of sampling ants: pitfall traps and baits. Pitfall traps consisted of a little 50 mil conical tubes I sunk into the ground, filled halfway with propylene glycol, diluted propylene glycol, left them out for 3 days for ants to fall into, to sample them passively. I also sampled foraging ants by putting out cookie baits for an hour in grids, and then would collect the ants by snapping Petri dish covers over them to look at whether or not ant abundance was related to the incidence of these spiders. I found no significant preference of the spiders to nest next to ant colonies versus not next to ant colonies. This graph is showing basically the average number of spiders per flag over the course of my survey, the 15 days I checked them, and there was no significant difference between ant colonies and non-ant sites in the presence of the spiders. So this graph is a little more complicated. So this is showing all of my 18 sites, field sites, where I set up grids to sample for spiders. Here they're numbered in by the abundance of adult ant-mimicking spiders from most to least abundant. This is the cumulative number of sightings in each plot over the course of all of the survey dates in that summer. So it reflects both the how many flags were being occupied and also how consistently. If they were just popping up once one survey and then disappeared, they wouldn't be counted as much as if they were consistently on those flags throughout the season. So this here is showing the abundance mean number of ants per pitfall trap in each of these sites, and this is showing the mean number of ants on the cookie baits in each of the sites. And what you can see here is that these bars here don't show any real relationship to the bars showing ant abundance in these sites. And there is indeed no significant correlation between ant abundance and spider incidence. So if site didn't have that many ants, it didn't mean it couldn't have a lot of spiders. So they didn't seem to be just picking up on sites with a lot of ants. I also use these surveys to assess habitat preferences of the spiders. So these bars are colored differently based on the type of habitat they represent. The orange are younger forests that tend to have a lot of light coming in, pretty well-developed understory of herbaceous plants. The white represents fields, which have pretty similar vegetation to the younger forest, just no tree canopy. And then the blue represents older forests that were much darker in the understory, had older trees, more closed canopy, and pretty sparse understory vegetation. And these are just showing 2 different years of surveys, and what I found is that the ant-mimicking spiders were much more common in both the field and the younger forest sites than in older forest habitats. They didn't really like older forests. You can't even see some bars here because they just were totally absent from some of these habitats. And this is just showing averaging across the different types of sites in those 3 categories. And that kind of fits with what has been known about their habits in Europe. They tend to like sunnier, warmer sites, so they don't tend to be found too much in shady areas. And given that, well, this is just an example of one of the young forest sites that you might find them in. This is in our arboretum. Pretty dense understory vegetation, a lot of goldenrod and other broadleaf herbaceous plants. This is generally a kind of opening in the canopy or forest edge. These are the kind of sites you don't find a lot of them in. This is actually, it's not really an old forest, but this has a thick canopy of European buckthorn, which is an invasive species in our forest and quite a problem. Blocks out a lot of light, and there's a lot of bare patches, not as well developed herbaceous understory. And this is an older forest. This is actually near my house where I have a bunch of our plots. We have an older forest behind us. Sometime one of our cats, Cinnabar, will join me on my ant survey, so that's why he's sitting there. And the understory is much more sparse. You don't have just a carpet of vegetation for them to forage in. Now, of course, they don't just nest on these vinyl flags. They must have some kind of natural nest substrate, so I was curious about where they would be going if they weren't nesting on my little flags. And as I started to look more carefully at plants throughout my field sites, I found that they indeed use leaves, which are very similar to the flags in the way they drape and fold and provide little places for them to attach their webbing to make these shelters. The shelters can get quite thick, especially when they're built by females that are producing eggs and raising as their spiderlings hatch. They might also be found in the leaf litter. This is a male that emerged from a little shelter just on a dead leaf on the ground. So those surveys were also helpful for understanding seasonal patterns in the abundance of these spiders. So these graphs, again, are showing the cumulative number of sightings. This is added up across all the plots at each survey point. So we're kind of averaging across all of the different areas sampled, but looking at the component of time from generally late May to early June to late September. Females were pretty consistently found on these flags throughout the whole season, but males were quite absent from the flags early in the summer and don't really start appearing until August. That doesn't mean they're not around necessarily. You actually do see people posting observations of these on iNaturalist, but they aren't actually using these flags that I'm using to survey as surveys for the spiders. This graph is showing different immature stages from eggs, which start appearing in kind of mid-June, to the spiderlings as they hatch, and then juveniles as they're emerging from their natal shelters and going off and building their own little shelters on the flags. And you can see this occurs over quite a broad range of time. I think maybe females are probably producing successive broods over the course of the summer. Often you find eggs along with spiderlings in the same shelter. And this is just showing some of these egg masses. They're little orange eggs. They're pretty distinctive. You can see they're usually in clusters of 10 to 15 in a little pile that you can see under the shelters. I often use a flashlight to help me visualize what's going on. I have a very bright LED light so I can see what's going on, movement of spiderlings. There's a little spiderling there. And the females actually stay in those shelters with the spiderlings and the eggs throughout the course of the season. The males, if you see that later peak of males and later in the summer is basically following this peak in juveniles. And often you'll start seeing the males being on shelters, on the flags, often with the juvenile spiders. Here's a little juvenile here, and a male emerging from the shelter, perhaps waiting for mating opportunities late in the summer. I could often see multiple spiders on a flag. There could be two females, females and juveniles sharing flags, or even two males. And I started to witness some of the interactions of these males on these flags, suggesting that there's competition for territory and access to mates on these desirable nesting sites. I'll show a little video that illustrates that. An interaction among some males. See, there's two males on the flag. They engage each other, do this mirrored dance. And then you see where those enlarged chelicerae come into play. Open them wide, show them off to each other, and then usually it ends with one of the spiders retreating, running away, and the other one standing its ground. Still waving its front legs around. Hanging out on the flag, taking up the territory. The other one just kind of disappeared down there. So one of my students, Jacqueline Zhao, was interested in looking at these male-male contests in more detail, what kind of factors determine which male wins the contest. We collected a bunch of males, and she measured them all and found a lot of variation in male body size, almost twofold variation in body length. And so she was curious if body size affects the outcome of these displays between males. And so she set up a little arena to pit one male against the other. She divided the males into small and large size classes and kind of randomly paired them in a number of different types of pairings, with small males matched with each other, small versus large, large versus large. There was always some variation in size between the males. Some were just more different in size than others. And I'll show you one of her behavioral trials. So the males, I think she acclimated to the arena with a barrier between them for a little while, and then removed the barrier and let them interact for 3 minutes. Eventually something happens. These are just 5 centimeter wide Petri dishes. See each other, engage in the dance again. You can see they also bob their abdomens back and forth. And then the chelicerae snap open. And that one decides it's not up to the challenge, runs away. And it didn't, continues to kind of avoid the bigger male for the rest of the trial. So Jackie ran a bunch of these trials with these different males of different sizes. And did indeed find that winners of these contests were significantly larger than losers. In basically 21 of the 25 contests, the larger male won. In the few cases where the larger male didn't win, they were very closely matched in size. So there could be other factors that are playing a role as well. You know, some of the other movements that they're doing might be playing a role in the outcome of these displays. So you might also wonder what these spiders gain from looking like ants. What are the benefits of their very close resemblance to ants in movement and appearance? And these spiders are not actually feeding on ants. They are, even though I see them in the vicinity of ant colonies, I've never seen them go into ant colonies. I've never found them in with the ants. So they're not using this as a form of aggressive mimicry to get in and prey on ants. So most people think that they are an example of Batesian mimics, and Batesian mimics are where a palatable species or a harmless species resembles a more dangerous or distasteful species in order to avoid predation. And ants are often thought to be undesirable prey items. They're often very aggressive. They can potentially sting you or are chemically defended. They also are not necessarily especially rich. They have a lot of chitin, maybe not the most nutritious morsel of food to get out there. So resembling ants might be a good way to avoid getting eaten. Just like this hoverfly, this harmless hoverfly resembles the dangerous wasp that can sting, even though it poses no danger to a predator. So I've had a number of students investigate the question of whether an ant-mimicking tree does protect Myrmaracity Formicaria from attack, as has been shown in other ant-mimicking spiders. Students Alana and Alice conducted trials where they pitted this predator, what we consider the predator, a larger salticid spider, against Myrmarachne Formicaria spiders, a non-mimic spider, and also ants. And they filmed these encounters for 3 minutes and recorded the outcome. So I'm going to show you some results, an example of one of those behavioral trials. Why did we choose this particular spider as our predator? One thing is just practical. This is a spider that's available when students are coming back in the fall and they can do some lab experiments. And it's actually a species that I frequently find actually using the flags. And sometimes they will even be on the same flag as the Myrmarachne, maybe one on one side and one on the other. So it's a potential enemy. They don't have a broad diet. So that's why we chose these particular species. So we're going to look at a series of trials. These are all the same predator individual paired with different opponents, not on the same day. These were separated in time. And all of their trials were like this, the same predator paired with a different opponent, and then they compared the outcomes among those different opponents. And here's one with the ant-mimicking spider. This one is actually quite energetic. Certainly getting the attention of the spider. It even attacks, but it rejects it right away for some reason. And did not attack again during the trial. Here's the same spider with an ant. This is a Formica glacialis ant, the most common ant species in my field site. Not attracting a lot of attention yet, but now it's moving around. So this case, it never attacked me again. Just kind of wandered around. Then we have the non-mimic spider. These weren't any particular species. They were just spiders that we could sweep and find at the time these experiments were done. They were all salticids of some kind. spiders were all at similar levels of satiation, and this did not survive this encounter. So across all of their trials, both ants and the ant-mimicking spiders were significantly less likely to be attacked by the salticid predator than the non-mimic spiders. At least half or less as often. Now, of course, you can notice that non-mimic spider was really small, and that could be part of a factor in the outcome of those experiments. So in the next year, my students, a different set of students, tried a different non-mimic spider that was more comparable in size to our predator. So maybe it wasn't just suffering from small size, making it more easy to attack. So we used, again, the same predator, Phidippus princeps that was used before, and the non-mimic was another Phidippus species, Phidippus audax. It's called the bold jumper. It's very common, very easy to find. It's often in crevices and wood piles. It comes into your house during the winter. So it was something we could collect of the right size throughout the fall. And so we didn't repeat the ant part of the experiment. It's pretty clear that the spiders didn't like to attack ants at all. I haven't looked at all the videos. A lot of these were done by the students. This spider, I don't think it is necessarily representative of all the mimics. This one's really got kind of an attitude. But it did not get attacked by the bigger salticid at all. So this is the species that we're using as our putative predator, and this is what we're using as the non-mimic. You can see there's much more aggression in these encounters. And so as a result, these are the results of their trials. The ant-mimics were much less likely to get attacked by the salticid predators than the non-mimic salticids. So it does seem like that Myrmaracity Formicaria is gaining some benefit from their resemblance to ants, at least in interactions with these spider predators. We have not tried other types of predators in these kinds of trials, though. So another thing that interests me was their overwintering behavior. You go out to these flags late in the fall, they're gone, they don't stay on the flags. It would be a very exposed area. They would freeze if they stayed on those. So where do they spend the winter? Some possible overwintering sites include leaf litter. I haven't actually found them there, but I have found them like opening up hollow stems, like raspberry stems, goldenrod stems, other things that might have hollow pith. And they can be found in there. Sometimes I find them dead, sometimes I find them alive. I've also found them in snail shells. So here's a little, you can see there's some webbing inside this snail shell that was on the forest floor. You can see the outline of a male's chelicerae there. And this has actually been documented in the Czech Republic that these will overwinter in snail shells in the litter, producing nice little refuges for them. They also use a lot of non-natural sites, things like yard debris, pots, and these seedling trays. These were left out on the forest floor from a seedling propagation effort. And you could find these shelters on their underside. And by June, females were laying eggs in these shelters. Wood piles are another place we found them. This is actually a remarkable log that we found in our wood pile. It had 40 different shelters of Myrmarachne on it. I marked them all and I tried to track them over time, and none of them survived, which was kind of sad. I did move the log to my own porch, and maybe I had disrupted the microenvironment. Or maybe there is just a really high overwinter mortality. It's hard to say. But we have recovered living spiders from our logs, things we bring into the winter, crevices, we find them over winter. This is actually a log that we found in January of this year. It actually had a male, a female, and a juvenile on it. All of them were alive. They could be revived. And the female, once I brought her into her lab, she laid eggs and produced spiderlings. So she was already mated and would have been potentially ready to start laying eggs in the spring. So all of the stages seem to show overwintering behavior. So finally, we're interested in kind of the history of this invasion of North America. How did these spiders get here? What are the dynamics of their spread? And one way we can try to get at that question is by looking at genetic variation in these spiders. Informed by iNaturalist observations where they've been cited recently throughout New York, Ohio, and Pennsylvania. We collected spiders in August of 2019, and kind of just like the iNaturalist pictures and what we've, our own experiences, we looked in a lot of kind of anthropogenic sites. This is actually a field station where they were first noticed back in 2001, so we went there. We did find them in places, you know, little crevices, like this is a gutter tray, and picked it up, and there's a Myrmarachne under that in a little shelter. Just, you know, odd places like this aluminum siding of this outbuilding. We actually pulled over because we saw a lot of flags. We were going to check the flags, but then we started seeing spiders all over the walls, and they're just climbing the walls, probably nesting in the vegetation against these. This is a nature center that had lots of pots, and we found in seedling trays, so we found them there. This is just another building next to a motel. So we were able to find spiders reliably in these different sites where they've been reported. Not all the sites, though, are these kind of odd areas associated with human activity. This is a spider found on the gravel shore of Hemlock Lake, which is one of the two undeveloped lakes, Finger Lakes in New York. So there's no shoreline development. There's very little foot traffic. You can only really get here by taking a boat. So, and I saw 3 spiders just along the shore here. So we sampled Myrmarachne from 14 locations in August 2019. My student Dan Fleischman extracted DNA from these spiders. And this describes our sequencing effort. First, we started with the cytochrome oxidase, a mitochondrial gene, which is commonly used for barcoding. And we found absolutely no polymorphism across all of our samples. And it's not necessarily that surprising. It is a barcoding gene that's chosen partly because it's not super polymorphic, so it can be used to identify at the species level. So we tried another gene, Adh Dehydrogenase, which is used, has been used to quantify intraspecific genetic variation in other spiders. My student Cassidy sequenced 27 samples of that gene. And this also showed very little genetic variation. Most of the sites, most of the sequences were invariant. There was a few minor substitutions. It was about 0.3% divergence. And this is much less variation than has been observed for other spiders for which this gene region has been sequenced. So that suggests that maybe there is really very little variation in this invaded introduced population. And that would be consistent maybe with a single introduction event, somewhere around the Great Lakes. It's thought that perhaps they came in on plant material or horticultural supplies, something like that. That happens a lot with spiders. But really, to understand the history of its spread, we need to maybe use more markers that are more reliably polymorphic. Also sample more widely. I don't have any Canadian samples. There could be, and the range is already spread even more. And also sampling in Europe as well to get some comparative samples to understand the genetic variation in the host, in the native range, and maybe even find out where they possibly came So we're working on developing additional molecular markers. My student Cassidy and I worked really hard to try to get some good RNA, and we're exploring RNA, what markers we can get from RNA sequence data as a kind of economical way of going about this. We haven't really investigated this aspect of these spiders, their phenotypic variation, but I take lots and lots of pictures, and I notice a lot of variation in their coloration. Everything from kind of a orangish chestnut color to more dark brown to almost completely black, with very little variation over their body in coloration. So why there is this phenotypic variation? Is it does it have a genetic basis? Considering we don't find a lot of genetic variation, at least the loci we looked at, who knows? But it's kind of interesting that there is this variation. There's also a lot of variation in the potential ants they could be mimicking or associated with, and so that's interesting to look at. There has been some documentation of more darker forms in Europe as well. So of course, I've been focused on this spider in North America, but that's not where it's from, and so I'm very interested in what it does in its native range. How do the native and non-native representatives of the species compare? Do they have similar phenological patterns? Do they have similar habitat preferences? As I pointed out, in North America, we find them in all of these settings where they're associated with humans, human activity, buildings, kind of sterile environments, not always with vegetation. Is that the case in Europe as well? Or is that some kind of new habit that they've developed kind of associated with their introduction probably into those kinds of habitats? Do they associate, what ants do they associate with? Are there commonalities with the ants that they are found among in North America? So partly to get at this, I had a student, Eliana Ontiveros-Oberg, kind of mine some of the data available out there, occurrence data for European representatives of Myrmarachne Formicaria. She looked at the Global Biodiversity Information Facility database. This is a database that compiles information from museum collections, biodiversity studies. It also includes iNaturalist data, not all the iNaturalist data, but data that's been more vetted for accuracy in its identifications. And this graph shows what she was able to find in terms of comparing seasonal patterns in Europe versus North America. We limited these data to just data collected since 2012 because that's when kind of the North America samples begin. And it is curious that you see this peak in North America in late summer, and that seems to be absent in Europe. They kind of have more of a peak in May, and not kind of are petering down more in the late summer and early fall. Whether this is a real seasonal difference in their phenology or just a collection bias, I don't know. We also looked at sex-specific patterns. Now, a lot of the records in GBIF don't have sex associated with them, but a lot of them do have photos associated with them, so we could look at photos, especially from iNaturalist, but also some other data bases. We couldn't necessarily be confident that we could identify a female, distinguish a female from a juvenile or subadult, but we could definitely always tell the males with those distinctive chelicerae. And there seems to be more kind of disparity between male and female patterns in North America, with kind of males peaking late in the summer. Then you see in Europe, there's not a lot of difference between males and females in Europe. This does kind of correspond to the peak I see in my data when I'm looking at my survey results. So, of course, I wanted to come to Europe to try to find this spider, to collect samples for potentially genetic analysis, to kind of see what kind of habitats it occupies, and what ants it's found with, all these questions that I had about what it does in its native land. So I gathered information from the GBIF database, iNaturalist data, some other resources like the German spider atlas, there's the Czech spider atlas, and also other sites that just seem to look like good habitat for them. This is showing all of the occurrences from the GBIF database. This doesn't necessarily represent a true distribution of the species because it's going to be very biased based on which countries are contributing to that database, how those samples get in them. There are actually 900 observations just from France. So we actually spend a lot of time in France searching. We didn't go to all of these sites, of course. But these are the sites that we did go to. So my husband and I came out in mid-July and to start surveying for these spiders. And this is kind of, we started out in Vienna, traveled through Czechia, through Germany, and into a bit of France, and then Belgium, and back into France, and all around back here to Spain, and then Italy, back into Austria. And through all these 90 sites, we only found 16 spiders. We didn't even find any Myrmarachne until down here in Spain. We also noted any ant-mimics, because we were always excited when we saw an ant-mimicking spider. So these are also shown here, Leptorchestes, Synagiles, and Myrmarachne. These are all salticids as well. So our first findings were other ant-mimics. This was our first find of an ant-mimic was in Lorsch, Germany, a site that was specifically found where Myrmarachne was actually reported from. But now I kind of wonder, there's a lot of confusion about these two spiders, so it might have been a misidentification to begin with. But they were all over this farmer's caravan, crawling along the walls. So, you know, juveniles generally, although I think I did collect a few adults there. Curiously, also all over this caravan, peeking out in the cracks were wasps. There were tons and tons of wasps among these spiders. So we collected them there. We actually did find one Synagiles by sweeping vegetation in this site as well. This is another site in Saarbrücken, Germany. There was an iNaturalist report near this area. So we're searching gardens. These are kind of places people find them, you know, finding them in the gardens is a popular thing. So people are working in their gardens, they find them. So we were looking around these garden plots, and I saw a little Synagiles running along the fence. And then we just stopped for lunch at a park, and my husband spotted this Leptorchestes on the playground equipment. So these are all kinds of places we might expect to see Myrmarachne just based on our experience in the States, these weird anthropogenic locations. These are just some close-ups of those other ant-mimics, Synagiles. This is actually the first ant-mimic to make it to North America. It's found around Montreal, and it's also found in British Columbia. But it has not spread to any great degree, although apparently it's very common in Montreal. What's curious about it is it raised its second pair of legs, like antennae, instead of its first. So if you look at them really closely, you can definitely tell whether you have this or another spider. But it takes some time looking at them. And then this is Leptorchestes, which does look a lot like a female Myrmarachne, so you have to carefully look at its profile to tell the difference. It has much more of a constriction in its abdomen, though. And this is a species, Leptorchestes berlinensis, that I'm working with Stano on some behavioral experiments here at Masquerading, because it's much more easy to get to than Myrmarachne, clearly, for any kind of experimentation. So our first success in actually finding Myrmarachne was in Spain. We almost didn't even go to Spain, but there was a few recent iNaturalist observations. I wanted to go to Spain because I speak a little Spanish, so it was one of the few places I could speak a little bit of the language. And we were pretty close in the Pyrenees. And I also communicated with Geoffrey here, who had done the post on iNaturalist, to actually say, could you tell me a little bit more about how you found it? And he actually came out and helped us look. And we actually found 10 spiders there. I netted one just sweeping vegetation. This is actually a hot spring, so people go for medicinal baths. He actually found it not because he was looking for spiders, even though he's always looking for spiders, but it just crawled up next to him on the bath So that's a kind of a typical thing for Myrmarachne, I think, for how people find it. And he was beating vegetation onto this pan, like these alders along the stream. And so we had, had found quite a few at this site. But they were all very small, so about 3 millimeter, tiny little juveniles. Then the next day, we went up to another place that was a locality that had reported Myrmarachne recently. We actually veered away from the actual locality because the landscape was just kind of parched at that point in August. And after we saw where we did find Myrmarachne, thanks to some water and some less vegetation, we knew we needed to find water. And so we found this river, the River Adh, and my husband Jeff was looking at this iris and saw a few little juveniles crawling on the plant. So I looked some more in there, and I actually was sifting in the litter, and I found one more that way. So we found 3 Myrmarachne there. And so we were always looking for alders and irises, and anything that was successful kind of informed our search from there on. We often would find multiple ant-mimic species in the same place. It seems, if you find one, sometimes you find the other. So this is a place we were sweeping in the brambles along this River Brenta in Italy, and we found both the Synagiles and a single little juvenile Myrmarachne. And these brambles are a place I would expect to see Myrmarachne. That's kind of the arboretum where I work in North America is just covered with raspberries, and they like those leaves for making their shelters. So everywhere we saw raspberries, I would sweep them, but to no success until here. And we finally found an adult at this pond on a trail near Bassano del Grappa in Italy. I think there was this little cottonwood, and it fell off of that cottonwood into Jeff's pan. It's very exciting to get an actual large adult male. So we had 3 total sightings in Italy. That same day, we found another little juvenile in a park in a town further north. And maybe I found one in Austria, but it's so tiny it could be a little Leptorchestes. I need to look at it with a better scope. So that was our great haul of Myrmarachne for the season. But so it's very curious. Meanwhile, we had to go back to New York to take care of some final long-term visa paperwork. I did one more survey in my field site. I looked at my 8 plots in the Arboretum, which I could go around to one day. And in one day, I saw 103 adult Myrmarachne spiders. 103 spiders, 60 adults. I also saw eggs, juveniles, spiderlings, every stage. On my garage door, it's just lined with shelters. You can go out and look up and down the garage doors. You have always a couple of spiders hanging around. So it's just night and day in terms of the patterns you see in Europe and North America. Yeah. So why are they so different? Why do we see such different patterns? There's some indications that they might show different phenologies. Maybe there's a shift in reproductive patterns in North America that is giving them more success, at least for now, as they spread throughout North America. We never really saw Myrmarachne in a very built-up, human-built setting like we see in North America. So maybe there's a shift in their acceptance of those kind of habitats. Or maybe there's some enemy that they're escaping, or some competitor, some disease, who knows? But for some reason, they're just thriving in North America, comparatively. Maybe they're around in Europe. My timing could have been completely off. As you saw in one of those figures, they seem to peak in May and June. Maybe they're just not as common in August. And why that is, I don't think it's necessary that it's any more hot or inhospitable in European summers than in North American summers, but who knows. The association with water is curious because my sites don't really have water. They can get pretty dry in the end of the summer. They're not near streams or ponds or things like that. So there are some interesting differences between these species, or between these populations. Of course, whenever you're dealing with an introduced species, you might be curious about what its ecological impacts might be. Is it having a negative effect on any of the fauna in North America? We do have some native ant-mimicking spiders. The only one I've seen around where I live is this salticid called Miramaracity Formicaria. It's a very delicate little spider, very long and thin. It produces shelters, but they're almost imperceptible. It will use my flags, so that's kind of how I've seen it more recently. Occasionally I'll see it crawling around in vegetation. There are some more ant mimics that are found further south and west of us as well. Of course, there's other spiders it could be potentially impacting, but it's hard to see how it could really be negatively impacting resources available to other spiders. There are some invasive spiders that have had negative impacts on native fauna in North America, but they tend to be web-building spiders that can potentially dominate habitat, displace other species more than I might imagine Myrmarachne could. But of course, it is something to look into. So finally, Miramaracity Formicaria provides a really unique opportunity to look at a species that's early in its colonization of a new environment, potentially encountering new interactions with different species, different ant fauna, different competitors, new habitats that maybe it didn't exploit in its native range, like these anthropogenic settings. And so it's kind of exciting to continue to pursue study of this species. So finally, I'd like to acknowledge many people who have helped with this research. For our experiments, we maintain a lot of spiders, and people have helped in caring for the spiders, like Ali Menendez, my husband Jeff Over, collection of spiders, Jeff Over, Adbiri, and Joffre Espigule Pons, who helped us out in Spain, the Geneseo Foundation for funding research and travel, as well as their support along with the Student Association for student research for many of their projects as well. And I want to thank all of my student collaborators that have contributed to a lot of the behavioral experiments, the genetic work, the data mining, Jacqueline Zhao, Alana and Alice, Dan, Cassidy, Julia, Naomi, Eliana, and some other research efforts that I didn't have time to share here. So I'd be happy to entertain any questions.
So thanks a lot for an amazing account of the biology and ecology of Miramaracity Formicaria, which is so rare here. I have to say that when Jenny contacted me that we could look for Myrmarachne, I was wondering where could we find it, because we had been searching for it for like 10 years, and I found maybe 10 individuals altogether. So then I more intensively went to the field, but we are no luck at all. So that's why, fortunately, we have some other ant-mimicking spiders that you can work here with, but not Myrmarachne.
Yeah.
So let's start the questions. Hope you have any comments or questions. Yes.
I have a question about the color of the flags. Because did you think about the possibility that they would prefer one against the other? For example, when you said that the female is staying near the shelter with the eggs, with the spiderlings, all over the summer. It also has to hunt, right? And I would expect, like we use yellow pants to catch insects, that maybe these yellow flags would attract more insects, so more prey than a blue flag. So did you consider this somehow in your
I did do a test to see if there were more spiders on one or the other, and there were not significant differences. But, and sometimes I've used other flags as well. I've tried just by sometimes I run out and I get another color. So pink, white, orange, they don't seem to necessarily show us a preference that I could tell. But I don't know if, I'm not sure. I don't know if, I've never thought about the other insects coming to the flags. It'd be interesting to look at. I mean, sometimes I will check the flags and there'll be a shelter there, and sometimes the female will be gone, and so she's out hunting away from the flag potentially and returning another time. So I don't, sometimes from week to week, sometimes you see the female, sometimes you don't, maybe depending on time of day.
This bright yellow color which you used, that's basically exactly, I think, the color which is often used to attract insects. You know?
Yeah.
So one would think that this might have an effect. Then just one comment. I mean, when I worked on a soil biological project in North America, I never saw so many European earthworms on 1 square meter than there.
Mm-hmm.
You know, so it is something about a fresh invasion.
Mm-hmm.
Usually they peak, and then the question is if this wave will ebb away or not.
Yeah, I mean, that's an interesting question. If they have an advantage now, that will eventually wane.
The color of the flag could be important also due to the thermal preference. That, for example, well, you have used blue, red, and yellow, right?
Blue, orange, and yellow. And
so, but if maybe the blue might be attracting more insulation or sunlight, so it might be warmer inside in the shelter than under the yellow one.
Yeah.
So this they could show some preference.
That'd be kind of interesting. We could try, maybe doing some choice tests with spiders would be interesting to try.
Okay. Yes.
So first when I saw the video that they are usually around ant colonies, I thought there must be as well like ant specialized, like eating on ants. But then you mentioned that they are actually not. So have you observed what they usually eat? Like, do they have actually like a preference? Maybe if you observed many times that they eat something, or is it whatever.
Yeah, I have not observed many cases of predation, but I've occasionally seen them with little gnats or flies, small flies, but mainly small flies is what I've actually, the kind of prey I've actually observed them with, when I have observed them. So yeah, but it's not something I see a lot.
You mentioned that ant-mimicking spiders sometimes protect the ants. Is it known why would they do it?
Why? I mean, potentially if they are adapted to exploit ants, it's a really rich, abundant food source if you can get away with it. But it's also a dangerous strategy, so I think there's a lot of trade-offs associated with it.
I mean, when the spiders defend the ants.
Defend?
Yeah.
Oh. I don't think they actually defend the ants.
But you mentioned that other species of ant-mimicking spiders sometimes can defend ants? Or do they...
Yeah, I don't think no. Sometimes they will prey on ants.
On the prey, don't they?
Yeah, but I don't think they defend them. So there's not any kind of mutualism that I know of. Interesting. Yeah, I don't think of any case like that.
I wonder what's known about the mating behavior. So if it's not mating, so you have the unique opportunity because you now have an access for an endless amount of spiders.
Yeah.
Maybe some cool stuff which is difficult to do here in Europe because they are very rare.
Yeah, I haven't really. We've actually tried doing some mating kind of trials in the lab. Like I had my students who were working on spider behavior this year, they were interested in, kind of which males female chose, and we would kind of we combine 2 males and a female in a little cage. We never really witnessed mating. Often, both males would pursue the females, and she'd just kind of run away from them. And so we haven't really. I've never actually definitely witnessed mating in captivity. So I don't know much about their behavior, but hopefully we can get more.
Wondering if females can feed on males if they have these huge mandibles. Maybe they can
Cannibalize.
well cannibalize. Like females mostly eat on males, but in this case the males have these huge mandibles, so maybe they can just handle the females better than the other spiders.
Maybe. Yeah, I've never actually seen. they've always been in captivity. They've always been basically civil to each other. I've never seen really cannibals in these cages, so I don't think they do that. I am curious, like with these males that are kind of hanging out on the flags, and there's a juvenile there. Do they actually know that juvenile is a female? You know, what kind of cues? You know, if you expose them to different silks from males versus females, are they attracted to those nesting sites that might have, you know, mean a prospect of future mating opportunity? I don't know, but there could be some interesting experiments you could do with that.
Okay, and the last one, I was surprised that they actually like cookies. Because I found cookies particularly annoying these days.
Ants,
yeah, and ants. You mentioned that you use cookies as a bait.
that is the ant people swear on pecan sandies as the cookie of choice to use for baiting ants, because I guess it's got the sugar, it's got the fat, it's got the protein from the nuts, so it's just it's just a kind of a standard ant thing.
Which cookie is that?
The pecan sandies.
Uh-huh.
So it's kind of a shortbread. The shortbread.
We don't have them here.
You know, you probably have something.
So everybody has except cookies, so even spiders.
The spiders are not attracted to the cookies.
Okay.
Yeah, just the ants.
Okay.
I have another question, and that's about these predation experiments.
Mm-hmm.
The ones where you use this larger, non-arm-mimicking predatory spider,
Mm-hmm.
which you had used twice in 2 different years.
Yeah.
But the second time, you took other, usually also jumping spiders,
Mm-hmm.
spiders, right?
Yeah.
Of a similar size.
Right.
So, you only compared, or you only talked about the situation where the species in focus was preying on the other one.
Yeah.
But didn't it also happen the other way around? I mean, what was the ratio of this spider being attacked by the other one, which was supposed to be the prey?
Right. Yeah. Why? It seems kind of arbitrary. My choice.
Yeah.
Well, I mean, part of the reason is we always use that one spider as a predator. We want to use that as the focal predator. But also the bold jumper, even though it's called the bold jumper, isn't as bold as the other spiders. So it tends to be I think we even did some trials using it as a predator the previous year, and it just kind of was not interested in preying. I think it was it might just been in overwintering mode and was not interested in
the one
attacking.
species which you tested against the ants and against Myrmarachne, that was the bold jumper, or that was another one?
No, that was
another one.
That was a different.
So in the second series of experiments, use this spider and always the bold jumper.
Mm-hmm.
Always, it was always the 2 species.
Yeah, yeah.
Not as in the first year, a mixture of different species.
No, it was always
okay,
it was always
one.
the black bold jumper.
And the bold jumper is not bold at all.
It's not that bold, at least not that time of year.
Okay, thank you.
Okay, any other question? Yes.
You mentioned there is another ant-mimicking spider which is not native to North America in Montreal.
Mm-hmm.
Are there even more cases of introductions or invasions of ant-mimicking spiders from other regions of the world? I mean, including tropics, Australia, Asia?
I mean,
I'm asking because...
yeah, is there something about ant mimicking?
Yeah, about the strategy of mimicking ants could be a kind of general advantage for spiders for invasions. I don't know what is the relation of non-native Untitled spiders which are non-ant mimicking and mimics make like more significant proportion than would be expected.
Yeah, I don't know. That's a good question. I can't think offhand. Other ones, and I think Myrmarachne has been found in other places besides North America, perhaps.
Other continents.
Yeah, and I think that's true for the other species as well. But I don't know for sure if other ant mimics have. But that's a good question. I could look into that.
Yeah. Maybe I missed it in your talk, but do you have some data about the origin of these non-native populations? Or are there any data about genetics of European populations that you can compare with mitochondrial variability?
No, yeah, we don't have any information to compare right now. Yeah. But so I don't know where it possibly came from. We actually stopped at a lot of nurseries to look for them, you know, and you know, look on plant material and pots and things, look for shelters. But unlike where, you know, the shelters we see all over various types of debris in North America, we hardly saw any kind of evidence of them like that. So, yeah, so I don't know.
To me, it seems like it would be very difficult to trace, to track different lineages moving or, well, spreading in North America, because probably with these anthropogenic habitats, nurseries and so on, they will be moved around by people all the time. So it will be all a big mix, I think.
Yeah, I mean, I think it's probably would be very hard to see if there's. I don't think they're moving around on their own volition.
Yeah.
I mean, like we've gone to places and we found an ant mimic in our car, and it probably came from our house, and then it could have jumped out somewhere, and you know, that's probably how it got to Chicago, and who knows. So I think they're colonizing things associated with humans and accidentally move them around.
And in this urbanized area where you found a lot of these species, are there near some ant nests?
Yeah.
Do you observe the ants in these environments?
I mean, I actually haven't seen those. Some of those sites I haven't actually gone to, so I don't know what they're like. I mean, you know, on our garage there actually are a lot of ants, so because we live in a rural area. It's, but in these more urban areas, it doesn't seem like there would be a lot of ants, and especially not like Formica ants. There could be other kinds of ants that tend to get moved around with people as well, which some of them could look a lot like in terms of the appearance of juveniles. Could look a lot like a Tamponoma, which is an ant species that gets around all over the place, especially in plants as well, plant material.
So what is the purpose, or what is the advantage for the species to be near an ant nest in natural context, in normal context?
Yeah, I don't really, I mean, normally it could just be, you might be more likely to be mistaken for an ant if you're in a bunch, with a bunch of different ants. Less, you know, a predator is going to find you harder to identify you as an actual spider because you kind of blend in. But, you know, in these other settings, maybe they don't really have predators, and they don't really need that. But they definitely don't necessarily require ants to thrive. Patterns in North America.
And I have another question. So in Europe, you found several times the species near wet environments. So do you think it's a pattern, or it was random, and because you were there, you found them there, or do they mind about humidity?
I think maybe it's more they need the plants to be healthy, you know, being among healthy vegetation where they might establish their shelters, have more insects to forage on, rather than kind of, you know, drying up in an open area where there's perhaps less prey and the leaves that they're sheltering on are withering. I don't know. I think it's more, I don't think they necessarily need water or need to be near a stream or attracted to those sites, potentially. I think it's more maybe the qualities of the vegetation, perhaps, and the prey opportunities.
In North America, you found the species in fields.
In fields.
And young forests, right?
In
so
forests.
In this kind of fields, I would expect that it's more like hot and dry.
It can be hot and dry, but there also can be some amelioration of the heat, you know, under the vegetation. If there's enough vegetation, and it doesn't get so dry that it dries up. But so I don't know. But they're not necessarily by the stream or by a pond in North America.
I have to say that most of the records from Czech Republic are from the vicinity of some water bodies, like ponds, lakes, rivers, and so that's very different from what you have observed.
Yeah.
So, any other question? Yes?
During your field trip, did you prefer or focus
Well, I don't think we went to really any old forest sites looking for ants, looking for the spiders. You know, we kind of went off the localities that we had information about, and then, you know, we get these GPS points, and we're not really sure exactly, you know, even how accurate they are. So we're looking kind of at forest edges, places where vegetation that's sort of similar to what I see at home, you know, brambles, broadleaf plants. I don't really expect to see them in like just grasses, because those leaves don't seem to lend themselves to the kind of shelters they would make. But also sunny areas, because you know, they need the warmth and light. So older forests, maybe not. You know, maybe they would be more common in like forest gaps, like a light gap in a forest. But then it's really hard for us, just not being unfamiliar with an area, to hike. Let's go hike in a forest until we find a gap, and you know, it takes a lot of time. So we're kind of just exploring, winging it. But I think we definitely were, you know, more in the field, forest edge type of realm of habitats that we were exploring, and then anthropogenic.
And did you see any difference between these, at least to younger forests and fields during your trip, or was it the same?
Yeah, I think everything. We probably didn't really find them in any true field areas. There were always kind of some trees around, and there's more shrubby, I guess, is more where we would find them.
These data on younger forests, older forests, they were from North America. They were from
Yeah. So
I'm just eager to know if the pattern is the same for the European countries.
Yeah, I think they're all kind of areas that had some woods, shrubby, and then herbaceous layers, but not necessarily big open fields. I think even though I swept a lot of those kind of areas, I did not find them. And I don't even, to look for them in North America, I don't even sweep for them. I just look for shelters. I look for evidence. You know, I haven't even really tried that. Although when I did get home, I did try the method that Geoffrey used, just beating vegetation over a pan. So I walked for 20 minutes down an edge of a forest in our yard, and I think I caught 10 in that 25 minutes. So maybe I will start using them. Because usually I just kind of look for their shelters, or I find them crawling around, or use my flags. So, but yeah, same things don't work here.
Any other question? Yeah, Michal?
So, while your Europe trip, you didn't find any spiders near ant colonies, if I remember it correctly. Did you look there, or like, would you think that in Europe maybe there would be other relationship between spiders and ants?
I mean, you know, if we were looking in an area and we didn't find any ants, we didn't feel very confident that there would be spiders there. Because I think if ants don't like it, then probably spiders aren't going to like it too much either. If we totally didn't find any ants and eating the vegetation. I did always collect ants with any spiders, and I guess there are kind of probably there are certain species, or not necessarily, I haven't identified them, but genera like Lasius I would find a lot with them, which the juveniles could look very similar to. I think the Formica is more something that would resemble the adults of the species, and I would see those in the vicinity. Generally, Formicine, there's this subfamily of ants that are in the Formicine, I think are more likely to be the model for these species, and another subfamily of ants in Myrmarachne. But I haven't actually tried to identify ants yet. But we did find ants with all of the places that we did find Myrmarachne. So ants crawling on the vegetation. But we also, you know, kind of used whether we find other spiders as a guide as well. So there's, you know, other spiders are often the same habitats as Myrmarachne, using the same flags. There's this spider, Enoclonathus, it's kind of yellow spider. Uses these big eggs. It's native to Europe, but it's in North America. So whenever I saw them here, I was like, well, maybe it's here because there's other spiders in this habitat. But
Did you check or know if there are any reports or any cases when people reported Myrmarachne in naturalists as an ant or as a spider?
I've seen an ant reported as Myrmarachne. Something that was clearly an ant. But none of, yeah, so, let's see.
They're mostly accurate. So if they report
Well, yeah, there is some mix up with Leptorchestes.
Okay.
Actually, I need to go through and add some corrections to those, because there's some that are being misidentified. But I don't know that, I mean, I guess there could be some ants. Yeah, I don't know. Well, yeah, that's possible. There's probably a lot.
To check and
Yeah,
see how people
yeah.
can be identifying.
I think the ant people would if there are people, so there's people who are mining, who are constantly checking these, that just do this as a hobby. They probably would catch that kind of thing, that kind of real severe difference.
They can count to 8.
Yeah, and just, other things, the eyes, But that's interesting.
So I actually wonder whether the success in the US and the success of the occurrence in the US is due to the enemy release. So could you please, if you consider the activity and the microhabitat specificity of Myrmarachne, could you name or list the potential predators?
I mean, other spiders kind of are the main things I would think of. I mean,
but the spiders have to be larger. Like some of the Philippus are larger, but not many of the spiders are larger than the adults.
And I mean, there's a lot of nursery spiders in those habitats, especially in the field areas. And I haven't tried any trials with those kind of things. There could be mantids. Maybe, I don't know if hemipterans would go after them. I even wonder about things like wasps as well.
How about some vertebrates, lizards or?
We don't have any lizards.
No.
Yeah, but we have no lizards where I am. Although we saw so many lizards in Europe, we wondered if in Europe could lizards be a major, because they're everywhere in these walls that we were looking for spiders because we thought they might be on these walls like we find in North America. We always see all these lizards and we thought, well, maybe they're eating them all. So lizards aren't huge. The only vertebrate we have flickers are a woodpecker that actually specializes on ants, and maybe they could get confused with spiders. But they're not going to be foraging. They forage on trails where ants are. They don't really go and pick off vegetation, pick in the vegetation. So.
And also the activity. Is Myrmarachne active during the mid of the day?
That's something I don't really know for sure. Because my surveys, I just go all day long surveying, so I don't go to any particular time. But there are times, they're not in their shelter. I don't know. But that would be something. That's something I'd like to do in the future. Is do more of a kind of diel pattern of the spiders. And how far do they go from these shelters? To forage. I don't know how I would track them, but it'd be fun to know.
So any other question?
Last one. Sorry about so many of them.
No, that's
but, in one of the experiments that you showed, that like Myrmarachne versus the larger jumping spider, so I saw the male using the mandibles against the predator. So I wonder how the female performed. You try to use the females if they were also resistant in the same way.
We haven't actually done experiments with the females because at that time of the year, usually when my students are doing these experiments, the males are just much easier to find than the females. So
I wonder if
it would be interesting. And I don't know, I haven't looked at all the videos. I'm not sure if they picked that one out as exceptional as a video. I'd have to look. I don't think all the Myrmarachne are that bold. A lot of them tend to just kind of run around. I'm also curious about to what degree they're using vibrations also as part of their mimicry. Because there's a lot of pounding and they're touching their abdomen to the ground and different things like that. there's so much to do with the species. And I'm learning, new methods working with Stano and his experience working with these behavioral experiments. I hope we can come back and apply to my abundant supply of subjects in North America.
Right. Any other question? I don't think there is any. So, thanks again. Of course, if you have some other questions some other time, you can meet with Jenny at our department, at our group, Building 8.
There's my email if you want to contact me. Yeah, thank you for all the wonderful questions and ideas and thoughts. Really great.
Okay, great.